Imagine for a second that you are sitting there celebrating a milestone birthday.
Right, like the big 5-0.
Exactly. You are blowing out 50 candles on your cake, and chronologically, you are exactly 50 years old. But what if, right at that very moment, you could look at a dashboard?
Oh, a biological dashboard.
Yeah, a dashboard that displayed the true biological age of all your internal systems. And what if that dashboard told you that, well, your birth certificate says 50, your heart is actually 40, your liver is 50, but your brain is already 60.
That completely shatters the way we are conditioned to view the human body,
Right. Because we always talk about aging as this single whole body process.
Exactly. We tend to think of it like the whole ship goes down the exact same speed. But I mean, the data we are unpacking today proves that it's just a complete myth. Our organs are aging on entirely independent timelines.
And we have an incredibly dense, fascinating stack of research to get through today to prove that. We are doing a deep dive into a groundbreaking new study from Nature Aging.
And it is a massive one.
Yeah, we are talking about organ-specific proteomic aging clocks. And like you said, this isn't a small trial. The researchers built this using data from over 43,000 people in the UK Biobank.
And then they actually proved it worked by testing it on massive global cohorts, right? Like the China Kadoorie Biobank and the U.S. Nurses Health Study.
So the mission for our deep dive today is to figure out exactly how your individual organs can age at completely different speeds. We are going to unpack how a single drop of your blood can reveal these hidden timelines and what it actually means for your future health.
Because this is basically the blueprint for the future of personalized medicine.
It really is. It's about understanding how the specific choices you make every day might be accelerating the aging of one specific organ without you even knowing it.
Or feeling a single symptom.
Right. So before we can look at which of your organs are aging the fastest, we really need to understand the mechanics here. Because, I mean, if my liver is sitting quietly in my abdomen, how can scientists possibly measure its biological age just by looking at a sample of my blood plasma?
Well, it really comes down to analyzing the proteins circulating in your bloodstream.
Okay, so proteins.
Right. And when we say they are looking at blood plasma, we aren't just talking about, red and white blood cells. Blood plasma is like this massive information superhighway.
Like a conveyor belt.
Exactly.
Yeah.
They carry thousands of different proteins that are constantly being shed by the tissues all over your body. And the researchers in this study used highly advanced technology, specifically a tool called the Olink Explorer 3072 panel.
Wait, the Olink Explorer 3072, what is that exactly?
It is a highly sophisticated assay. Basically, it allows them to measure nearly 3,000 distinct proteins circulating in the blood all at once.
Wow, 3,000. But right, since blood goes everywhere, if you just have this giant soup of 3,000 proteins, how on earth do you know which ones came from the liver and which ones came from, like, the lungs?
That was the real breakthrough. They had to trace those proteins back to their origin. So they cross-referenced all this blood data with massive tissue databases.
From the GTX database, right?
Right. Yes, exactly. The Genotype Tissue Expression Project. By using GTX, scientists mapped out the origins of every single protein. And out of those thousands of circulating proteins, they found 418 that are what we call organ enriched.
Meaning they only come from one place.
Pretty much. They are highly specific to just 10 major organ systems. We are talking about the brain, heart, lung, immune system, artery, intestine, liver, kidney, muscle, and pancreas.
So these specific proteins are overwhelmingly produced by just one of those organs and nowhere else.
Exactly. They act like highly localized location beacons.
I sort of think of it like a master mechanic analyzing a car.
Oh, I like that analogy.
Yeah, so the blood plasma is basically like the oil stream that is shared by the entire vehicle. If that mechanic drains the oil and finds traces of a very specific metal alloy, an alloy that the manufacturer only uses in the transmission gears, they immediately know the transmission is grinding down.
Right. Even if the engine itself looks totally fine.
Exactly. That specific metal in the oil tells a highly localized story of degradation.
And taking that car mechanic analogy one step further, finding the metal in the oil is only the very first step, right? The mechanic still has to determine if that amount of metal means the transmission is just broken in, or if it is on the verge of total failure.
So having the organ-specific proteins is really just the raw data.
Exactly.
Right.
The researchers needed to figure out which patterns of those proteins actually correlate with the true aging process. And to do that, they turned to artificial intelligence.
This is where the methodology gets really wild to me.
Right.
They didn't just use standard statistics or look for simple high or low thresholds, did they?
No, they used a very complex machine learning model. It is called LightGBM.
LightGBM, what does that do?
It is a gradient boosting framework. Essentially, it is an AI that is incredible at handling complex, nonlinear data to separate the true signals of aging from just random biological noise.
But how does the AI know what is noise and what is a real signal?
Well, they used a technique called Baruda feature selection.
Baruda, okay,
Yeah.
break that down for me.
Basically, the researchers fed the AI the real blood data, but they also injected fake randomized data alongside it. Decoy proteins, essentially.
Oh, wow. So they tried to trick the AI.
Yes. They forced the algorithm to mathematically prove which real protein signatures were actually better at predicting age than the completely fake ones.
That is brilliant.
It is. They trained this system on tens of thousands of people teaching it to recognize the exact microscopic patterns that correspond to the chronological age for each of those 10 specific organs.
Okay, let's pause for a quick recap of where we are, our sort of 5-minute milestone, because this is the foundational mechanism for everything else we were going to talk about today.
This is good.
First, our blood holds specific proteins uniquely tied to specific organs. Second, researchers trained advanced AI models like LightGBM and Baruda to read these protein levels and calculate a biological age for 10 different systems. And third, this works consistently across massive, diverse populations in the UK, China, and the US.
Perfect summary. Which brings us to the absolute core concept of the entire paper, which is the organ age gap.
The organ age gap. What is the technical definition of that?
It is the mathematical difference between your chronological age, the number on your birth certificate, and your organ's proteomic age, which is what the AI calculates from your blood.
So if I have a positive gap,
That means accelerated aging. Your organ is biologically older than you are. And they found people who had what they called extreme agiotypes.
extreme agiotypes.
Yeah, these are individuals whose organs are aging vastly faster or slower than their peers, like more than 1.5 standard deviations away from the norm.
Okay, I have to play the skeptic here for a second. Let's say I already have a diagnosed kidney disease. My kidneys are failing. Obviously, my kidney proteins are going to be out of whack, right? The AI is going to say my kidney is old. Isn't this just a really expensive way to diagnose a disease I already know I have?
That is a very valid question. It is called reverse causality, but the researchers specifically controlled for it by conducting a prodromal analysis.
Prodromal, that means pre-symptom, right?
Exactly, the pre-symptom phase. It is the window of time before a disease is clinically diagnosed or even noticeable to the patient at all. They weren't just looking at sick people. They look at people who appeared completely healthy.
So the clocks can spot this stuff early.
Yes, the clock spot accelerated aging years before the disease actually sets in.
Give me an example from the data.
Let's look at Alzheimer's disease. They examined individuals who were perfectly healthy at the baseline of the study. They passed all their cognitive tests. They had no memory issues whatsoever. But years later, they went on to develop Alzheimer's.
Okay.
When researchers look back at their blood from that baseline healthy period, those individuals already had a brain age gap of 2.2 years older than average.
Wow, 2.2 years older. That is a brain quietly crying out for help. But wait, what was the rest of their body doing at that exact same time?
That is the crazy part. Their overall organismal age gap, the average of all their systems, was only 0.5 years older.
Wait, really? Just half a year?
Just half a year.
That discrepancy is terrifying. The body as a whole looks relatively fine. The patient feels totally normal. But their blood is sounding a massive alarm that the brain is aging more than 4 times faster than the rest of the organism.
It completely reframes how we view cognitive decline. And it highlights why the brain clock is arguably the most critical finding in this entire paper.
More than the heart or the liver.
Oh, absolutely. The brain clock didn't just predict neurodegenerative diseases like dementia. Out of all the organs they measured, the brain clock was the single strongest predictor of all-cause mortality.
Wait, the biological age of your brain is the best predictor of when you're going to die from anything.
Yes, from anything. And furthermore, it was a massive driver of multimorbidity.
Multimorbidity, that is getting hit with multiple chronic diseases at once, right?
Exactly, both physical and mental. The brain age gap uniquely predicted physical multimorbidity, like developing heart disease and diabetes simultaneously. But it also predicted neuropsychiatric multimorbidity, like severe depression and anxiety occurring together.
So if the brain starts aging rapidly, the rest of the biological network, Just follows it off a cliff.
Basically, yes.
Well, since the brain clock is this master predictor for when we get sick and when we die, we desperately need to know what drives it to tick faster.
Right. What is causing it?
Yeah. Are we just victims of our DNA? Like, are some people just born with a timer that runs out faster, or is it our choices?
The paper proves it is a deep combination of both. On the genetic side, they used something called a GSS, a genome-wide association study.
Which scans the entire genome.
Right. They wanted to see which variants strongly correlate with accelerated organ aging. For the brain, they found highly significant links to a gene called GBR1.
GBR1, what does that gene actually do?
It controls GABA receptors. GABA is an inhibitory neurotransmitter. You can think of it as the brain's brake pedal. It calms the electrical signaling down.
Okay, so if you have a faulty GBR1 gene...
The brakes are inefficient. The brain essentially overworks itself. The signaling gets chaotic. And because of that constant overdrive, the brain physically ages faster.
So there is a hardwired component. But you mentioned lifestyle, too, which is the good news. It means we aren't completely helpless.
Far from it. The data show that a poor diet, smoking, and particularly a lack of sleep directly accelerate specific organ clocks.
I really want to dig into the sleep aspect. The paper highlighted the pancreas and the brain taking a hit from bad sleep. Why would missing hours of sleep literally age the physical structure of the brain?
It comes back to the proteins we were talking about earlier. Think of sleep as a literal wash cycle for the brain.
A wash cycle?
Yeah. During deep sleep, the brain actually shrinks slightly. This allows cerebrospinal fluid to rush in and literally flush out toxic proteins and metabolic waste that build up during the day.
Oh, wow. So if you constantly shortchange your sleep...
That wash cycle never finishes. The trash builds up. And that buildup causes deep, chronic neuroinflammation.
And the AI is actually seeing the results of that trash buildup in the blood.
Exactly. The AI relies on very specific proteins to calculate the brain's age. For instance, one protein is called NEFL.
NEFL, what is that a marker for?
It is a marker of neuroaxonal injury. It basically only shows up in the blood when the physical, structural cables of your neurons are snapping and leaking out.
That paints a grim picture. What else?
There is another one called GFAP. This one spikes heavily when the brain's support cells, the astrocytes, are highly inflamed. So the biological clock isn't just some vague metric of oldness.
It is literally detecting structural brain damage.
Yes. Structural damage and cellular inflammation years before you ever forget your keys.
You know, I found the connection between the brain and the arteries to be one of the most illuminating parts of this whole deep dive, because they didn't just look at the brain in a vacuum.
No, everything is connected.
Right. The AI highlighted arterial proteins, things like ELN and LTBP2.
Ah, yes. Those are extracellular matrix proteins.
Extracellular matrix, explain that for us.
It is basically the cellular scaffolding that keeps your arteries flexible and fan. When the levels of ELN and LTBP2 change in the blood, it tells us that the scaffolding is stiffening and degrading.
I visualize it like a house.
Okay, how so?
You have the plumbing, which are your arteries, and you have the electrical wiring, which are your neurons. This paper physically demonstrates that the plumbing and the wiring degrade together.
It is a great way to put it.
If the scaffolding of your pipes stiffens up, the pressure changes. Eventually, the drywall protecting the wires, which in the body is the blood-brain barrier, starts to leak.
Exactly. And once that blood-brain barrier breaks down, the inflammation kicks in.
The electrical wiring shorts out, and those structural cables of the neurons start snapping, releasing those warning proteins like NEFL into the blood.
Right. Early vascular degeneration closely mirrors the disruption of that blood-brain barrier. It is actually a hallmark of early Alzheimer's pathophysiology. The AI is literally tracking the sequence of your house breaking down.
And speaking of Alzheimer's, we have to talk about the APOE4 variant, because anyone who follows this stuff knows APOE4 is the notorious Alzheimer's risk gene.
Yes, if you carry it, your risk skyrockets.
Yeah.
It is the genetic hand nobody wants to be dealt.
But the researchers found something incredibly profound about APOE4 in this study, right?
They did. They looked at people who carried this dangerous genetic variant, but who had managed to maintain a super youthful biological brain age.
Meaning their brain clock was ticking much slower than their chronological age.
Yes. And having that super youthful proteomic brain age appeared to grant them immense resilience to the APOE4 variant.
Wait, really? It canceled out the risk?
It dramatically lowered their risk of dementia compared to what the genetic models predicted. It's a massive revelation because it suggests that biological age can trump genetic destiny.
Wow. So if you keep the biological age of the organ young, you can potentially override a severe genetic predisposition.
Exactly. Through lifestyle, diet, or future interventions, you can fight back against your DNA.
Okay, let's do our 10-minute milestone recap because the implications here are just huge.
Let's do it.
We have established that organ age gaps predict future diseases, not just existing ones. We know the brain clock is the master predictor for mortality and multimorbidity. Both genetics, like the JABR1 gene, and lifestyle choices drive this aging. And finally, vascular degradation and brain aging are deeply intertwined at a molecular level.
The mechanisms are undeniably clear from the data.
They are. But here's where I get stuck on the practicality of it all.
Okay, what is the sticking point?
It is incredible that scientists can track all of this using 3,000 proteins and massive supercomputers running late GBM AI algorithms. But that is pure research.
Sure.
My local doctor struggles to get my routine cholesterol results back on time. There is no way a standard clinic is going to run a 3,000 protein panel on me at my annual physical.
You are absolutely right.
If it requires a supercomputer, it isn't a medical tool I can actually use. How does this become a reality?
Well, the researchers were acutely aware of that bottleneck. Clinical translation, getting this out of the lab and into the real world, was their ultimate goal.
So what did they do?
They applied a mathematical technique called recursive feature elimination.
Recursive feature elimination sounds like they are trimming the fat.
That is exactly what they did. They essentially asked the AI to play a game of survival. They said, if you could only keep the absolute most critical proteins and you had to throw the rest away, which ones hold the most predictive power?
So they forced the AI to simplify its own test.
Yes. They started with the 70-plus proteins that made up the brain clock, and they mathematically shaved it down to just 10 proteins.
Just 10.
Just 10. And for the overall organismal clock, which started with hundreds of proteins, they reduced it down to just 20.
But surely cutting out thousands of data points destroys the accuracy of the clock? I mean, you are losing so much information.
You would think so, but it didn't. The 10-protein brain model retained over 88% of its predictive accuracy for dementia and mortality compared to the massive full model.
That is astonishing. Over 88% with just 10 proteins.
And the 20-protein overall clock retained almost 89% of its performance. By distilling the signal down to its absolute most potent core, they created a test that is incredibly feasible for standard clinical laboratories.
Right, because 10 proteins is something a local hospital can actually handle.
Easily. It completely bridges the gap between sci-fi research and a routine blood draw.
Okay, let me push back on that one more time, though.
Go for it.
If the goal is a simple test for a doctor's office, why reinvent the wheel? We already have standard clinical tests. Blood pressure, cholesterol, fasting glucose.
Right, what we call phenotypic age.
Exactly, phenotypic age. Why do we need a new 10-protein blood test when we already have a blood pressure cuff?
The researchers ran a direct comparison to answer that exact question. They pitted their new proteomic clocks against standard clinical biomarkers and established phenotypic age models.
And who won?
The protein clocks vastly outperformed the traditional tests.
But why? I mean, blood pressure and glucose are major indicators of health.
They are, but they are macro-level symptoms. High blood pressure or elevated glucose is the end result of a biological system that has already failed.
Ah, I see.
Proteins, on the other hand, capture the root cellular mechanisms. The proteomic clock is detecting the loss of synapses, the activation of inflammatory cells, the stiffening of the vascular matrix.
So if we use the house analogy again.
Yeah, let's go back to the house.
The phenotypic age, the blood pressure cuff, tells you the roof is currently leaking and the floorboards are warped. The house is already damaged.
Exactly. But the proteomic age tells you the termites have just moved into the walls.
It sees the microscopic cracks in the foundation years before the macro symptoms manifest.
And that window of time is everything in medicine. It is the difference between prevention and damage control.
It fundamentally changes the paradigm of growing older. We are moving away from this monolithic, standardized idea of chronological age.
Right. It is no longer just, I am 45 years old.
It is this highly precise, localized understanding. Like, my heart is 40, my liver is 50, and my brain is 42.
By listening to these unique, distilled protein signatures in our blood, we can track exactly how our specific systems are degrading.
Which is incredibly empowering. It removes the guesswork. If you know your kidney is aging rapidly while you are still in that pre-symptom phase, you have time.
You have a runway to fix it.
Right. You can change your diet, alter your hydration, or use targeted pharmacology to slow that specific organ's clock down before chronic kidney disease ever takes root.
It moves medicine from reactive to truly proactive.
But it also opens up a Pandora's box of societal and personal questions.
Oh, absolutely.
We have just spent this deep dive exploring how a simple 10-protein blood test could soon tell you exactly which of your internal organs is secretly aging the fastest and precisely how close it is to failure.
It is a lot of power.
So if you could walk into a clinic tomorrow, give a vial of blood, and get that dashboard, would you actually want to look at it?
That is the million-dollar question.
And take it a step further. Think about how this reshakes society. If biological organ age becomes the new undeniable standard of health, what happens to the world around us?
Like the financial sector.
Exactly. What happens to life insurance policies when the underwriter can see that your brain is biologically 10 years older than your chronological age?
Or retirement age.
Right. What happens when retirement is no longer based on the year you were born, but the proteomic vitality of your heart and lungs? Does knowing your specific organ-level biological expiration date empower you to seize control of your health? Or does it just terrify you?
It is a heavy thing to think about.
It is. We started today thinking about blowing out 50 candles on a birthday cake. But the reality we have uncovered is that your body isn't celebrating one birthday.
No, it is celebrating dozens of them.
Dozens of them, on entirely different timelines, hidden deep within your cells. The science to read those timelines is officially here. The only question left is, are you ready to read the invitations?